Today we'll be talking about the history of virology. Like we discussed last week, virology is the scientific study of viruses and the infections they cause. And for years, these viruses have been with us, but they only started getting noticed in the closing years of the 19th century. We're going to be talking about scientists that made one contribution or the other towards the discovery of viruses and actually finding vaccines for the cure of those viruses. The first person we'll be talking about today is Louis Pasteur. He lived from 1822 to 1895, and he actually worked with bacterial cultures like the one that causes chicken cholera and anthrax. But while he was working on them, he discovered something about immunology. He observed by chance that old bacterial cultures lost their virulence. He instructed an assistant to actually inject chicken with a fresh culture of the viral bacteria before an holiday. The assistant, however, forgot and went on vacation. When he returned a month later and performed the procedure using the old cultures, unexpectedly, the chicken only showed mild signs of the disease and survived. When they were healthy again, Pasteur injected them with fresh cultures of the bacteria, and the chicken did not become ill. This led to the discovery of vaccines. The notion of using a weakened form of the disease to provide immunity was not new, but Pasteur was the first to take the process to the lab, impacting all virologists who followed after him. The microbe weakened in the lab had taught the chicken immune system to fight the infection without causing any serious harm to the chicken. This type of vaccine is called a live attenuated vaccine. Pasteur continued to explore illness in pursuit of new vaccines. And so when rabies became a challenge in 1885, although Pasteur did not know it was a viral infection, but he was able to actually produce a vaccine that was able to curb the rabies infection. After successfully protecting dogs from the disease, Pasteur agreed to treat his first human patient, a 9-year-old boy who had been seriously bitten by a dog, and there was little doubt that he would die if nothing was done. This was, however, able to save the boy, and the boy did not develop symptoms. This procedure was originally called Pasteur's treatment, but Pasteur decided to honor the 18th century virology pioneer Edward Jenner, who publicized the cure for smallpox, and give this artificial weakened disease the generic name of vaccines. The next person we're going to talk about is Charles Chamberlain, a colleague of Louis Pasteur. He lived from 1851 to 1931. The invention that allowed viruses to be discovered at all was the Chamberlain-Pasteur filter. Chamberlain produced a filter that could allow only viruses to pass through and retain every form of bacterial infection on it. It consisted of unglazed porcelain candles with pore sizes of 0.1 to 1 micron, that is between 100 and 1000 nanometer, which could be used to completely remove all bacteria or other cells known at the time from a liquid suspension. Though this simple invention essentially enabled the establishment of a whole new science, virology, the continued development of the discipline required a string of technical developments. The next scientist we're going to be looking at is Martinus Beijering, who lived from 1851 to 1931. In 1876, Adolf Meyer at Wageningen Agricultural Experimental Station in Netherlands was first to show what he called tobacco mosaic disease as being infectious. He thought it was actually caused by either a toxin or a very small bacterium. But later in 1892, Dmitry Ivanovsky used a Chamberlain filter to study what is now known as the tobacco mosaic virus. His experiments showed that the crushed leaf extract from infected tobacco plant remain infectious even after filtering it. Therefore, Ivanovsky suggested that the infection might be caused by a toxin produced by bacteria. However, in 1898, the Dutch microbiologist Martinus Beijering became convinced that the filtrate actually contained a new form of infectious agent. He observed that the agent multiplied only in living cells, and therefore he called it contagium vivum fluidum. That means a soluble living germ. And this reintroduced the word virus. Beijering maintained that viruses are liquid in nature, a theory that was later discredited by the American biochemist Wendell Meredith Stanley, who proved that they were in fact particles. The next set of scientists were two friends that worked together, Frederick Loeffler and Paul Frost. In 1898, they passed the first animal virus through a porous clay filter and discovered the cause of foot and mouth disease, FMD, in cattle. Loeffler and Frost described their discovery that the foot and mouth disease agent was neither a known bacterium or a toxin, but an ultra-visible, ultra-filtrable substance, which had actually gone unnoticed due to its small, minute size. In the extensive and precise experiment undertaken, they were able to transmit the disease from cows suffering from foot and mouth disease to other susceptible animals by transplanting germ-free filtered vesicle nymph. The vesicle nymph is like liquid that forms inside a blister in the foot of these animals. Now they took the fluid and filtered it and injected it into other animals and found out that these animals became sick. Loeffler and Frost were able to rule out a toxin as the cause of FMD by serial transmission of filtered vesicle from diseased animals, according to the meticulous calculations which they made. If the cause of FMD was toxin based, then after several animal to animal transmission, the original material should be so diluted that only one of the 2 trillion of the starting substances will remain. a toxic effect of that nature would be unbelievable, they concluded. Now, since the disease ran its characteristics course with every round of transmission, the causative agent therefore must be some sort of minute agent capable of reproducing itself. Loeffler and Froch reported that cow and sheep can be artificially immunized. Immunization was achieved by injecting the filtered vesicle fluid, which has first been sufficiently preheated to destroy the infectivity, or by injecting a vesicle lymph immune blood mixture, or passively by the serum of the immune curve. Loeffler and Froch were therefore the first to use an inactivated vaccine to fight a virus infection. In view of their contribution, Loeffler and Froch can justifiably be regarded as the founders of animal virology. The next scientist we are going to talk about is Wendell Stanley, who lived from 1904 to 1991. He was a young chemist working in Rockefeller Institute laboratories, and he purified a virus in the form of needle-shaped crystals with chemical properties of a protein. It was perhaps the most startling discovery to date in the Institute's history. How could a virus with its ability to infect and multiply also be an inanimate chemical? This prompted the discussion of life, the discussion of the question, what is life? As well as other criticism whether viruses are living or non-living. Further research elsewhere confirmed in Stanley's laboratory and soon demonstrated that the infectious substance was in fact a combination of protein and the nucleic acid. The result called attention to the similar reproductive powers of viruses and genes and helped lay the groundwork for modern molecular biology. Stanley actually experimented with tobacco mosaic viruses, which cause spots on the leaves of infected plants. By the time he began his studies in the early 1930s, TMV was well known to laboratory researchers. Stanley decided to apply to TMV method for crystallizing protein that have been developed by his Rockefeller colleagues, John and Moses. Although more than 300 human, animal, and plant viruses were known to science, when Stanley began his research, it was not known whether viruses were in his world, either inorganic, carbohydrate, hydrocarbon, lipid, protein, or organisms in nature. In addition to opening questions about the basic nature of viruses, it set the course for the field of virology. Stanley's own research had a more immediate impact on medicine, such that during the World War II, he turned his attention to developing an inactivated vaccine for viral influenza. He used a Sharple's centrifuge, a piece of equipment widely used in the dairy industry at that time, to obtain concentrated and purified quantities of the virus. The centrifuge's large capacity and efficiency made it possible to actually scale up production of vaccine, and Stanley's method was used in the development of commercial vaccines. Okay, the next person I'm going to be talking about is an English bacteriologist, Frederick Twort. He lived from 1877 to 1950. In 1915, Frederick discovered the action of bacteriophages on staphylococci bacteria. He noticed that when grown on nutrient agar, some colonies of the bacteria became watery or glassy. He collected some of these watery colonies and passed them through a Chamberlain filter to remove the bacteria, and discovered that when the filtrate was added to fresh cultures of bacteria, they in turn become watery. He proposed that the agent might be an amoeba, an ultramicroscopic virus, living protoplasm, or an enzyme with the power of growth. It is interesting that he was actually attempting to grow Yersinia virus in culture, and that it was a contaminating staphylococcus that he noticed was being lysed by his infectious agent. The next scientist we will talk about is Felix de Herel. He lived from 1873 to 1949. He was a French-Canadian microbiologist, and he discovered an invisible antagonist when added to bacteria on agar could produce areas of dead bacteria. The antagonist, now known to be a bacteriophage, could pass through a Chamberlain filter. He accurately diluted the suspension of viruses and discovered that the highest dilution, that's the one with the lowest virus concentration, rather than killing all the bacteria, formed discrete areas of dead cells. Counting these areas and multiplying by the dilution factor allowed him to calculate the number of viruses in the original suspension. He was the one that made a major contribution in the estimation of viral numbers. He realized that he discovered a new form of virus and later coined the word bacteriophage. The Herel discovered different types of bacteriophages that could infect several other species of bacteria, including Vibrio cholera. Bacteriophages were heralded as a potential treatment for diseases such as typhoid and cholera, but their promise was forgotten with the development of penicillin, and this has led to a renowned development of penicillin. But since the early 70s, bacteria have continued to develop resistance to antibiotics such as penicillin, and this has led to a renewed interest in the use of bacteriophages to actually treat serious bacterial infection. Now, the last but not the least, we're going to be talking about Dennis Bucket. He lived from 1911 to 1993. He was born in Ireland, and he was the first to describe a type of cancer that now bears his name, Bucket lymphoma. This type of cancer was endemic in Equatorial Africa and was the commonest malignancy of children in the early 1960s. Now, in an attempt to find a cause for the cancer, Bucket sent cells from the tumor to Anthony Epstein, and with his other colleagues discovered that viruses that resembles herpes virus were in the fluid that surround the cell. The virus was later shown to be a previously unrecognized herpes virus, which is now called Epstein-Barr virus. Why it can cause such a devastating illness in Africa is not fully understood, but reduced immunity to viruses, which they are suggesting is caused by malaria, might be blamed. Epstein-Barr virus is important in the history of viruses for being the first virus shown to cause cancer in humans. The first animal virus was the second virus ever discovered and is known as the foot and mouth disease virus in farm and other animals. This was discovered by German scientists Friedrich and Frost. Again, their sterile filtered liquid proved infectious in calves, providing the first proof of viruses infecting animals. The two went further by showing that it was possible to vaccinate cows and sheep against the disease using filtered vesicle extract that has been heated sufficiently to destroy its infectivity. This was possibly the first use of an inactivated virus as a prophylactic vaccine. Also, the first human virus is the yellow fever virus. This probably originated also in Africa and was spread along with its mosquito vector, Aedes aegypti, to the Americas and the neighboring islands by the slave trade. The virus was discovered and reported in 1901 by the U.S. Army physician Walter Reed after pioneering work in Cuba by Carlos Finley, reported in 1881, hypothesizing that mosquitoes transmitted the deadly disease. The agent became the subject of intense study because in the Spanish-American War in Cuba in the 1890s, about 13 times as many soldiers died of yellow fever as died from wound. The subsequent eradication of mosquitoes in Panama is probably what allowed the completion of the Panama Canal installed because of the death rate among workers from yellow fever and malaria. Now, the second half of the 20th century was the golden age of virus discovery, and most of the 2,000 recognized species of animal, plant, and bacterial viruses were discovered during these years. These discoveries have continued in the 21st century as new viral diseases such as SARS and Nipah virus have emerged. Despite scientists' achievements over the past 100 years, viruses continue to pose new threats and challenges to man. This is the end of the class. I need you to read about these scientists that have made contributions to virology. And in your spare time, I want you to bring out in not more than 3 bullet points the contribution of each of these scientists to virology. I need you to read and please do this assignment. Thank you. See you next week.